Okay, good evening everyone, good evening. Okay, good evening everyone, good evening. Okay, good evening. So friends, let's see in the morning class, I hope every one of you quickly recalled. We have started discussing about the immunogenicity, antigenicity, as well as the antibody structures, as well as the functions. So friends, in this particular class we will discuss about the another important topic from the immunology. Good evening everyone. That is the hypersensitivity. So friends, this is also very important topic, friends, where the questions were asked oftenly. Hypersensitivity. Friends, please listen. In this particular, there are only two ways of asking question from this particular topic. That is the factual statements, then followed by, friends, experimental. Okay, these are all the only two ways that we can expect the questions. Okay, so good evening everyone. So friends, please pay attention. What is the difference between the allergy and hypersensitivity? Everyone listen to me very carefully, friends. Allergy as well as the hypersensitivity. Good evening everyone, good evening. So friends, in one simple word, everyone please remember me, in one simple word, the severity of the allergic reaction only we can call it as the hypersensitivity. In simple word, remember the definition. What I am saying is, what friends, the severity of the allergy means, when the allergic condition is not properly treated, then that allergic condition will give rise to the hypersensitivity conditions. Kindly don't get confused with this particular point. Okay, see friends, everyone listen, all the students try to understand the points. Typically the inflammatory responses rid the body of antigen and resolve the infection within days. See friends, while studying the inflammation, I have told to you many times, friends, why does the process of inflammation has to occur in our body? I have told you many times, the process of inflammation has to occur in our body. The ultimate goal is what, friends? To prevent. What to prevent, friends? We already studied. To prevent the spreading of infection. To prevent the spreading of infection, the inflammation has to occur. What will happen when the inflammation occur? That particular pathogen will be restricted only to that area. Suppose example, friends, this is the area inflammation occur. So this is the healthy area and this is the healthy area. So here pathogens are there. What I am saying here, suppose example, if the inflammation occurred here, friends, that particular pathogenicity or the infection, good evening, it never ever spread to the healthy areas. That's the reason the process of inflammation has to occur. You all people know very well, when we get a small injuries during the time of playing sports, what will happen? That particular area immediately get inflamed. We all people know that very well, correct na? So friends, that particular type of reactions we can call inflammation. In certain cases, what is happening, friends, everyone pay attention, listen to me carefully. In some cases, the inflammatory response can have a harmful effects. Even, friends, it leads to the death also. Suppose example, there is a inflammatory responses spread through the blood. So there is a severe inflammation in the blood cells. The infection is completely spread throughout the blood. So of course, friends, that is a very dangerous reaction, undoubtedly. So that kind of dangerous reaction only we can call it as a hypersensitivity. This type of immediate reaction is called as a hypersensitivity or allergy. In a simple way, severity of allergy, severity of inflammation, we can call it as, friends, hypersensitivity. And every student has to remember one common point. What is the common statement every student has to remember? Hypersensitivity exclusively mediated by whom? Friends, please remember, I will explain one by one. If the hypersensitivity wants to occur, compulsory there will be a requirement of the antibody and antigen complexes. Antibody and antigen complexes are very compulsory in the formation of this particular hypersensitivities. From where we can expect the questions, friends? We can expect the questions from these four types. Surprisingly we can get the question from this particular slide itself, whereas in a given matching question, in one column they will ask all the type of hypersensitivity. Good evening Sema. In one column they will ask all the kinds of hypersensitivity. In another column they will ask the major causes why that particular hypersensitivity is occurring. So according to the two scientists Gell and Coombs, the hypersensitivities are of four types. I don't know what you do, friends. It's your responsibility to remember all the hypersensitivities without fail. Type 1 hypersensitivity, remember it is called as, friends, IgE mediated hypersensitivity. Don't get confused. Actually we should not use the word IgE mediated hypersensitivity. Actually that is a wrong statement. We should always use the word that elevated IgE level. What is the statement we should use? Elevated IgE level will causes this particular type 1 hypersensitivity. Please don't get confused. And friends, next one is what? Type 2 hypersensitivity. Type 2 hypersensitivity, friends, we can call the antibody mediated hypersensitivity. Follow me very correctly, friends. I am using the word antibody mediated hypersensitivity, but I am not at all saying which antibody is mediating the type 2 hypersensitivity. So we can use the word any. We can say that it is a generalized condition. But friends, when it comes to the type 1, it is extremely specific. It is extremely caused by the elevated level of only immunoglobulin A, not any other. Now look into the picture. See, one type of hypersensitivity, always try to compare with the another type of hypersensitivity, friends. By that we can remember it easily. In case of a type 1 hypersensitivity, it is specifically with respect to the IgE, and type 2 it is antibody mediated. But what is the common in the type 1 and type 2? It is with respect to antibody. But friends, when you look into the type 3 hypersensitivity, what is the statement you have to remember? It is both antigen and antibody complex mediated. Don't get confused with this particular point. What, friends? Type 3 hypersensitivity is, it is formed by the combination or the complex of both antigen as well as the antibody. And there is a specialized one, type 4 hypersensitivity. Friends, please keep it in your mind. Type 4 hypersensitivity we generally call it as the DTH. What is DTH? Delayed type of hypersensitivity. Now what is the reason why we are calling it as the delayed type of hypersensitivity? The reason is very simple, friends. There will be an involvement of the many cells. Since there is an involvement of the many cells, so activation of all the cells is not a simple process. It takes a greater time. That's why we are calling it as the delayed type of the hypersensitivity. Do not get confused with this particular point at all. Okay, and I am explaining in the form of a diagram. Friends, even you can get the diagrammatic based questions also here, which means they can give the 4 diagrams and they can ask a question that which of the following diagram depicting or which of the following diagram representing a specific type of the hypersensitivity. Even, friends, the questions can be asked in that way also. So all the students pay attention. See, look into the picture. This is what cell? All the students listen. Just think it, either it is a mast cell or the basophil. Even in the morning class also we have studied, friends. Mast cell or basophil, on its surface they are having what kind of a receptors, friends? They are having the receptors for the IgE molecule. We all people commonly know that. Now listen to me, friends. Whenever we have exposure to the allergen, what happen? The IgE antibody will come and attaches or reacted with that particular allergen. then it will allow the process of degranulation. In the next slide, I will explain you how does the degranulation occur very specifically. Just we are reading in this particular slide, friends, the overview. Remember in your mind, this particular type 1 hypersensitivity is causing exclusively because of the very high levels of the IgE. That's the only point you have to remember. And I have told to all of you that the type 2 hypersensitivity is caused by the antibodies. But the point is very clear. Which antibodies involved we cannot predict here. In the type 2 we can generally say that type 2 hypersensitivity basically causes ADCC reactions. I have told to you very clearly, last time also we have studied antibody dependent cell mediated cytotoxicity. In simple words, the type 2 hypersensitivity cannot take place without the involvement of an antibody. All the students, please look into the diagram very carefully, friends. This is the cytotoxic T cell. What cell it is? Cytotoxic T cell. All the students pay attention. On the cytotoxic T cell, there is a receptor is present. Focus, friends. And that receptor is associated with what, friends? You can see, right? That receptor is associated with the antibody. Now, friends, all the students look into this. These are all what, friends? We can call the antigens. The surface antigens that are present on the targeted cell. Now, friends, antibody will come and attaches to that particular surface antigen. Then antibody stimulate the cytotoxic T cell. Then the cytotoxic T cell will show the process of the cytotoxicity, which means that the cytotoxic T cell is going to destroy this cell. Simple. Now, same way, these are all the surface antigens that are present on the targeted cell. Now, friends, to this particular surface antigen, who will come and attaches? Complement molecule attaches. But the point is very clear, friends. Does the complement molecule is attaching to this very directly? No. Friends, all the students pay attention. The complement molecule attaches to this with the help of a specific antibody. So what is the common statement you have to understand? Whether it is the action of a, listen my friends, follow me correctly, whether it is the action of a cytotoxic T cell, whether it is the action of a complement molecule, what it will be requiring? It will be requiring the antibody. So what you have to understand, in case of a type 2 hypersensitivity, there will be a compulsory involvement of the antibody. That is what exactly we will be calling it as the ADCC, antibody dependent cell mediated cytotoxicity. So in the type 1 there is an involvement of antibody, type 2 there is an involvement of antibody. But friends, when you look into the type 3, all the students please follow me correctly. In the type 3 there is a formation of a immune complex. Friends, when we use the word immune complex, we cannot say that the immune complex is only consist of an antibody or only consists of an antigen. What we can say that the immune complex is the combination of both antibody also, friends, as well as the antigen also. Don't get confused with that particular point. Okay. Now, friends, all the students look into the diagram. This is the neutrophil, correct na? Friends, follow me very correctly. We have studied many times in the blood circulation also. Neutrophils are the cells which is going to first arrive at a site of inflammation. What, friends? Neutrophils are the cells which arrive at the site of inflammation first. Now the question is very clear. How do what is the property of neutrophils to enter into the damaged area? We all know very well. I told you many times. I'll explain you once again. We can call the extravasation. What, friends? Extravasation. Extravasation is the process of entry of the neutrophils into that particular damaged area. So how does this neutrophil or the extravasation is happening? that extravasation is happening just because of the immune complex. So in the immune complex, what are all involved? In the immune complex, complement system is involved. In the immune complex, antigen is involved. In the immune complex, friends, antibody also involved. Please do not get confused with that. So the last and final type, friends, type 4 hypersensitivity. What do you have to remember? Delayed type. You only look into this. First, there is an activation of the macrophage. Macrophage releasing certain cytokines which are activating the T cells. Then T cells get activated. Then the T cells activate the B cell. Then B cell differentiated into plasma and memory. Then finally, friends, the plasma cells are going to synthesize the antibodies. We all people know that. And the point is very clear. This process is not an easy process. And one more important point, that process never takes very faster. This process will take a greater period of time. That's why we can call it delayed type of the hypersensitivity. So friends, when you are revising after this class, you all people give the highest importance to the type 1 as well as the type 2. From these two only, most of the time we can expect the questions. Okay, so friends, now I am going to explain you one by one very briefly as well as the very elaboratively. Okay, focus. It's important, you have to remember it. Majorly focus on the mechanisms. Type 1 hypersensitivity. See friends, allergens. You never call every antigen as a allergen. So certain antigens speciality is what? There is a no need to be parasitic in nature. Even though they are non-parasitic in nature, they are having the ability or capability to stimulating the type 1 hypersensitivity. An example, let's take a pollen grain. Does the pollen grain is a parasitic in nature? Is it a parasite? No, not at all. Pollen grain is a completely non-parasite. But what's the speciality of the pollen grain? It can easily constrict the bronchi and it will cause great difficulty in the breathing. So most of the time, follow me very correctly, most of the time the allergens which are, follow me friends, most of the time the allergens which are causing the type 1 hypersensitivity, they are non-parasitic antigens. Please remember that particular statement. Now friends, what do you mean by the word atopic individuals? You have to remember, I can use the word 2 points. Atopic individuals means who are having the genetic tendency to that particular allergen. In other words, who are mostly sensitive. You know, right? When we shift all of a sudden from very hot climate to a very cold climate, what happened? Some people can easily adjust the condition, but some people will become continuously running, sneezing. There's a difficulty in breathing also. So what is happening here? They are having the genetic tendency. In other words, they are extremely susceptible, sensitive. They are super sensitive to that particular allergies. So the people, follow me friends, the people who are having the genetic tendency, or are the people who are more sensitive, those kind of individuals only, friends, we will be calling them as the atopic individuals. What we will be calling them as atopic individuals. Don't get confused. And what's the point you have to remember? In this particular atopic individuals, friends, there is a regulatory defects in the IgE. Now, the word regulatory defect doesn't mean that the IgE level is drastically dropping down. That's the wrong statement. IgE levels are extremely higher. What friends? IgE levels are extremely higher. Please don't get confused with that particular point. See friends, the next 3 to 4 slides are extremely important slides. Next 3 to 4. I am requesting all of you pay utmost attention and try to understand the mechanism very, very clearly. See, you all people know that we already studied how does the T helper cell and B cell can interact, and many times I have explained you with the help of a BCR receptor, TCR receptor, with the help of a CD4 and interleukin molecules. Now every student remember, once the T cell interacted with the B cell, what happens? B cell get activated, then B cell differentiated into plasma cell as well as the memory cell, and we all know very well that that plasma cells are going to synthesize this particular antibodies. Once the plasma cell synthesizes the antibodies, anyway, since we are talking about what? Allergy, we are talking about the hypersensitivity. So what is the statement every student has to remember? You have to remember the antibody synthesized here is undoubtedly, friends, IgE. See here, we have written a very nice statement. What is the nice statement we have written? Allergen specific IgE. What we have written, friends? We have written a statement that allergen specific IgE. Follow me very correctly. Now this allergen specific IgE, all the students understand, they are in order to associate to the master cell. See here how many receptors they are having, all we can call it as FC receptor, which are very specific. They will come and attach us. Now you only try to understand, friends, the moment IgE attaches to the receptor. Listen, follow me very correctly. See here how many granules are present in the master cell. There are many granules are present in the master cell. Friends, all these granules are getting breakdown. This is only we can call degranulation. Now for the exam point of view, what we people has to read very correctly, very briefly I mean to say, you have to study. Imagine this is a master cell, this is the receptor. What is this receptor? Called as FC receptor, and to this FC receptor who is attaching? IgE attaching. After the attachment, what is happening? The process of degranulation is happening. Correct na? This thing we have to study very briefly and very elaboratively now. So what we have to study for the exam point of view, follow me friends, pay attention, be patient. We have to understand what is FC receptor. We have to understand how does the FC receptors are associated with IgE. How does the process of degranulation is happening? I mean to say, friends, we have to read the mechanism involved in that degranulation. All the students listen. If you look into the FC receptor, there are two type of FC receptors are there. Focus everyone. FC epsilon R1 receptor, FC epsilon R2 receptor. Friends, there are total two type of receptors are there. And what is the point every student has to remember? The major point, you have to remember that FC epsilon 1 is said to be, follow me friends, is said to be high affinity immunoglobulin E receptor. Don't get confused. It doesn't mean that FC epsilon R2 do not have the affinity. It is also having the affinity, there is no doubt about it, but most of the time IgE, immunoglobulin E, in order to show their mechanism, most of the time it will be associated with the FC epsilon R1 receptor only. Kindly do not get confused with that particular point. Now look into the picture carefully. If you look into this is entirely FC epsilon R1 receptor. It is having different domains. Just remember, you have to remember the structure, that's all. It is having alpha domain, it is having beta domain, it is having the gamma domain. And friends, let me tell you one unique statement. When you look into the alpha domain, it is having large extracellular domain. Correct na? You can see here. And if you look into the gamma domain, friends, if you look into the gamma domain, what they are having, if you look into the gamma domain, it is having large cytosolic domain. Correct na? If you look into the beta domain, they are having a very short extracellular domain and they are having short cytosolic domain. Anyway friends, we cannot differentiate the alpha, beta, gamma. Listen my statements. This is what? This is called FC receptor. I am talking about this entire thing is only we can call the FC receptor. But I am just saying that this FC receptor consists of a alpha domain, beta domain, gamma domain. Whether any domain is a shorter domain or a larger domain, it doesn't matter. All alpha, beta, gamma together only they can form the complete FC receptor. That's the statement you have to remember. And friends, please no need to remember about the FC epsilon 2. Most of the time it cannot be used by the IgE. That's why we can call low affinity IgE receptor. Okay, now every student has to understand this particular slide very elaboratively. If you understand this particular slide very elaboratively, friends, the concept is very clear cut. Okay, all the students pay attention. This is what I am explaining to you. Mast cell, nothing but FC epsilon R1 receptor. Listen, FC epsilon R1 receptor, to which IgE will come and attaches. This is what I am going to explain you now. And I am going to explain how does the breakdown of the granules occur, how does the degranulation is taking place. Listen, I'll explain twice, just pay utmost attention towards the concept. It's easy. What is the first point everyone has to remember is our goal. Our goal of this particular pathway is breaking down of the granules, which we generally call it as the degranulation process. So we have to understand how does the process of degranulation is happening. Okay, now all the students pay attention. Listen to me very carefully, don't confuse. Follow me correctly. This is IgE, right? Now friends, IgE cross-linked with the allergen. Follow me friends. IgE cross-linked with the allergen, or IgE is associated with the allergen. The moment IgE associated with the allergen, what is happening? There is a activation of the FC epsilon R1 receptor. I'll explain twice, don't worry, follow me. There is a activation of FC epsilon R1 receptor, that is alpha domain, beta domain, gamma domain. All the domains are going to get activated. Then it is going to activate an important kinase, which is the PTK. You have to remember that. Protein tyrosine kinase enzyme is getting activated. Friends, the moment protein tyrosine enzyme get activated, it is going to stimulate the phospholipase C. We all studied very clearly in the cell signaling also 2 days back. Once there is an activation of the PLC, what we have studied, friends, PLC, that is phospholipase C, it is breaking down what we studied clearly. It is breaking down the PIP2. It is breaking down the PIP2 into IP3 and DAG. Friends, please don't get confused. What I am saying now, first PTK get activated, PTK activate the phospholipase C, phospholipase C activate the PIP2, phospholipase C break down the PIP2 into DAG and IP3. All the students look into the diagram. Now IP3 will helpful in bringing the stored calcium from the endoplasmic reticulum. So calcium came out. Now what is the function of this particular DAG? Everyone please listen my statements very carefully. Follow me correctly. The DAG and IP3 together, what they are going to do? They are going to activate an important protein, which we will be calling it as, friends, protein kinase C. This can be activated. Follow me friends. Now you know what is going to happen. Now this particular protein, see, what is my goal? My ultimate goal is I have to break down the granules. That's the ultimate goal. Now friends, this particular protein kinase C, what it is doing, friends, it is just breaking the granules. That's all. Sorry, it is just pushing the granules. Anyway, let me explain in a simple way. All the students listen. Suppose example, all the granules are present here. Example, imagine all the granules are present here. Now, if the granule wants to break down, follow me friends correctly. If the granule wants to break down, what has to be happened? These granules has to come and fuse the membrane. Yes or no? Of course, yes. The granules has to come and fuse the membrane. If the granules wants to come and fuse the membrane, granules has to move. The granules has to move. How does the granule movement occur? That is the question. See here, all the students look into this. These are all the granules. Now the granule starts moving. How? Just because of rearrangement. Rearrangement of what? Rearrangement of the microtubules. And friends, rearrangement of the microfilaments. Very, very simple point it is. Rearrangement of the microtubules and rearrangement of the microfilaments, they will allow the granules to move near the membrane. Then my friends, please listen my statements. With the help of a chemicals called as, follow me friends, fusogens. What is that? Fusogens. So they will help the granules to fuse the membrane and they will start the process of degranulation. Once all the chemicals will be released, obviously it will cause the allergic or hypersensitivity reaction. Very good. There will be a rearrangement of, correct Sushma, actin, microtubule and microfilaments. Mostly microtubule and microfilaments can undergo the rearrangement. Friends, I'll quickly summarize. Whomever did not understand, just pay attention. This is antibody IgE and these are all the FC epsilon R1 receptors. First, what is happening? I will explain one pathway, then I will explain the second pathway. So friends, first the allergen cross-linked with the IgE, then IgE activate the FC epsilon R1 receptor, then alpha, beta, gamma domains of FC epsilon R1 receptor get activated. Then FC epsilon R1 receptor going to activate a protein called as a protein tyrosine kinase enzyme. That enzyme is going to stimulate the phospholipase C. That phospholipase C is breaking down the PIP2 into IP3 and DAG. So what the IP3 is doing, friends? IP3 is going to release the calcium from the smooth endoplasmic reticulum, and DAG and calcium together activate the PKC. And friends, what this PKC is doing now? Now this particular PKC will come and will cause the rearrangement of the microtubules and microfilaments. What it is doing, friends? It is causing the rearrangement of microtubule and microfilaments by that the granules will come near the membrane. With the help of a chemicals called as a fusogens, the process of degranulation occur. This is the first mechanism of degranulation. Friends, the degranulation can also occur in the other type of mechanism. Same, all the students pay attention. Allergen will come and attaches to the FC epsilon 1 receptor. The moment allergen will come and attaches to the FC epsilon R1 receptor, what is going to happen, friends? Every student follow me very correctly. Same, protein tyrosine kinase will be activated. Here, friends, follow me. Here, what the protein tyrosine kinase is doing, it is going to activate. Everyone remember, friends, just compare, that's all. In the first case, follow me correctly. In the first case, protein tyrosine kinase activated the PLC. What it activated? It activated the PLC. Here it is just easy to remember. In the second case, protein tyrosine kinase is going to activate the phospholipase A2. That's the point I want you to remember, nothing else. Now, this phospholipase A2, it will helpful in the breakdown of phosphatidylcholine. First, we can't produce the phosphatidylcholine directly. All the students follow me. The phospholipase A2 first convert the phosphatidylserine to the phosphatidylethanolamine. Then the phosphatidylethanolamine, with the help of a PMT2 and PMT1, you don't need to remember that, it is converting into phosphatidylcholine. Friends, all of you keep it in your mind. This phosphatidylcholine will do two functions. One is what? Phosphatidylcholine is going to activate the fusogens. We discussed it, right? What's the function of a fusogen? The major function of the fusogen is the degranulation. That is the first function. And the second function of the phosphatidylcholine, very important function. Phosphatidylcholine plays a crucial role in the breakdown of the arachidonic acid. I don't know what you do, you have to remember this. Whenever phosphatidylcholine break down the arachidonic acid, what is going to happen, friends? It is going to give rise to the leukotrienes as well as the prostaglandins. Don't get confused with that particular point. And I told to all of you many times, in our body, if the process of inflammation occur, that inflammation occur just because of the prostaglandin only. Please don't get confused with that. Leukotrienes are the one which can cause the pain also, right? So this is how the degranulation occur as well as the inflammation also occur. Now you have to get a valid doubt. What is the valid doubt you have to get? You have to get a valid doubt that sir, does these two mechanisms, are they occurring step by step, or are these two mechanisms occurring simultaneously? This is the valid question every student has to get it. Now the answer, they never occur step by step. Both pathways occur simultaneously. Both pathways occur at the same time. Because then only, friends, the process of breakdown or the process of degranulation occur very faster. Don't get confused. I hope everyone see right now what I have explained to you is only this. Just I have explained you this. How does IgE associated with the Fc epsilon R1 receptor, how does IgE is involved in the breakdown of the granules or the degranulation? We understand very clearly with the help of mechanisms. Okay, now the question is very clear. Now the granules got broke down. Yes or no? What, friends? Now the granules are broke down. Now the question is, what kind of mediators are present in the granules? This is also important with respect to the part B. There are two type of mediators are there. Primary mediators are there, friends, as well as the secondary mediators are also there. Every student follow me very correctly. What do you mean by the word primary mediators? Keep it in your mind. Listen my statements. Whether the process of degranulation is occurring or not, follow me correctly. Whether the process of degranulation is occurring or not, friends, actually they will be stored in the granules. That's what we can call the primary mediators. Example, friends, histamine, proteases, as well as the heparin. These are all the major primary mediators. And friends, listen my statements very carefully. Secondary mediators. What do you have to remember about the secondary mediators? Listen to me. They are produced only during the process of degranulation, which means that if the process of degranulation never takes place, the secondary mediators are also never takes place. I hope in the previous slides only I told you by the mechanism 2 what is getting released. Leukotrienes are getting released by breaking down of the arachidonic acid. Prostaglandins will release. Platelet activated factors will be released. We can call PAF. So they will be released only during the process of degranulation. Whereas these can be stored before the degranulation. That's why we can call primary mediators. Don't get confused. And friends, follow me very correctly. What is the major difference between the localized anaphylaxis or the systemic anaphylaxis? Anaphylaxis is a type of a shockic reaction. It's a type of a shockic reaction. And anyway, friends, the name itself indicating, right, localized anaphylaxis, which means that it is extremely specific for one particular tissue. An example, let's take an example, friends, allergic rhinitis. When we take an example of the allergic rhinitis, what is happening here? Running nose. Right. Now, what is the point you have to remember? This allergic rhinitis, it is a very specific to the nose or the nostrils. It is not with respect to the any other part of the body. If you look into the asthma, why this particular asthma is happening? Inflammation, severe inflammation of bronchioles or the constriction of the bronchioles. That's what we can call it as the asthma. Now that asthma is not with respect to the every part of the body. It is only with respect to the bronchioles. Now atopic dermatitis, you know very well, on the skin surface there is a type of water bubbles will occur. If you break down that bubble with a force, the water oozes out, liquid oozes out, and there is a chance that it may spread the infection everywhere. So that atopic dermatitis, it is also what? It is also very specific to the skin. In the three condition, what is the common point? The reaction, whatever is taking place, that reaction is very specific to the particular tissue only. That's what we can call the localized anaphylaxis. Then friends, what is systemic anaphylaxis? Shock like a fatal state, which is including all the part of the body. Best example, all the people remember, when some people take the seafood, when some people will take fishes, they are super allergic to that particular seafood. The moment they eat the fish, within 20 to 30 minutes, in some people it is very fast, within 10 to 15 minutes, they are all over the body, lot of rashes will appear very fast. Correct na? Because they are sensitive to that particular food. And let's take an example, snake bite. When the snake bite occur, the venom of the snake, not only snake, the venom of bee also. What is that? It is a highly proteinaceous in nature. It can spread all over the body. It can reaches to the central nervous system. It collapses the central nervous system. It follows, then later it will attack on the blood circulatory system, which leads to the paralysis and eventually leads to the death also within the hour. Why? Because it is not restricted to one particular part. It is completely restricted to all over the body. That is, friends, we will be calling it as the systemic anaphylaxis. Don't get confused with that. Okay, now so far we have very clearly understand what we understand so far. What is type 1 hypersensitivity? How does the type 1 hypersensitivity will occur? What is the mechanism of the degranulation? That is what we people studied nicely. Now we have to understand the methods to study the methods to understand the allergies, the methods to detect the allergies. There are many methods are there, friends, to detect these allergies. Everyone just pay attention. Skin test, okay, injections or scratching, local. See, I will tell you, these tests are okay, but these tests are not at all accurate. I want every one of you understand my question. This skin test is an actually older method. This skin test is only a older method. Follow me very correctly. What is this method? You know, suppose example, all the students pay attention and listen what I am saying. So this is my hand. On my hand, at one particular area, exactly at this particular area, follow me friends, exactly at this particular area, there is an allergic reaction occurred. Okay, now before I am talking about olden days, not olden days, previously, when we visit the physician, you know what they will do? Suppose, look into this diagram, everyone pay attention towards the diagram here. Suppose example, this is the area where the allergy occurs. This is the allergy, what I am drawing with the red color. Now what the physician will do in order to understand because of what this allergy occurred. Because of what this allergy has been occurred, you know what they will do? They will start applying various type of allergens on our skin. Follow me very correctly. In order to understand how does this particular allergy occurred, they will treat us with a lot of allergens. See here, 1, 2, 3, 4, 5, 6. They have added lot of allergens. All the students listen. Suppose example, I got the allergy because I exposed to the lot of grass. Now you know what happened? That entire area, there is a accumulation of the mast cell, severe inflammation will occur. Then the people will confirm. People will confirm that whatever the allergy that you got just because of the grass. But this is not at all an accurate method. Now let me tell you one interesting point here. What is the interesting point? Listen. Here I got allergy. Now the physician tested me with all these allergens. Finally, the physician understood that the allergy was not at all caused by any of this. Any of this. Now what is happening? Understand the concept very correctly. Finally, we understand that the allergy is not caused by any of this. After few days, you know what will happen? Most of the people will show the sensitivity towards any of this. As a result, what happened? The new allergies starts developing. What friends? New allergies starts developing. So that's why we don't use this particular skin test at all. And why previously they always used to prefer the skin test are because of which is quick as well as the inexpensive. You can observe the accumulation of the mast cells very faster. That's the reason only, friends. So we will observe this, otherwise we don't observe it actually. We don't prefer also. Now I think you might heard the news. I think two months back in Uttar Pradesh there are 14 people. There are just 14 people got HIV. And the point is here, they did not participated in any unprotected sex. They did not participate in any unprotected sex, and they did not undergone any blood transfusion also. Then how the 14 people got the HIV positive? The simple, the method how it got spread is all the 14 people went to the same tattoo shop. When they visited the same tattoo shop, that particular person start using the same needle for everyone. See, a very minute mistake, because he is using the same needle for all the 14 people. This needle was previously used by user for one HIV positive person. So from there, we know very well the HIV can only transmitted through the blood as well as the vaginal fluids only. So what is happening through that blood? It was spread to the all 14 people. So always remember. So I am not saying that I'm not saying that tattoos are dangerous or not dangerous. What I am saying is when we are going for the tattoos, make sure that we will always use the sterilized needles and very fresh needles, fresh needles, and we will always use the unused needles only. Okay, don't get confused with that. Okay, now as I am saying that, friends, the skin tests are not a kind of accurate test. They are not at all accurate test. Now what are the accurate tests nowadays we are following? All the students listen, don't get confused here. We will use two tests to understand the allergies. One we can call, friends, wrist. Another one we can call it as the RAST. Okay, don't get confused. Wrist represents radio immunosorbent test. RAST represents radio allergosorbent test. I will give you two simple definition of this. Don't confuse my words. Suppose example, in my body, follow me, everyone follow me very correctly. In my body there is a allergy occur. In my body there is a allergy occur. When there is a allergy or the hypersensitivity reaction occur, what happens in my body? In my body, undoubtedly IgE level will be rising. What is happening in my body? There will be an increased level of the IgE. Correct or not? Yes. So friends, by using wrist method, what I can observe, all the students follow me correctly. By using wrist method, I can easily understand. What I can easily understand? Whether the particular person having elevated IgE level or not. IgE levels increased in his body or not, I can understand by the wrist. Okay, that's fine. We understand that in that particular person body, IgE levels are very high, which means that in that particular person there is a lot of allergic conditions are there. Now you all people know that we confirmed, what we confirmed, the person having allergy, but we never know because of what allergen. Follow me correctly. Because of what allergen, because of what substance the allergy occurred. Because of what substance or because of what allergen that allergy is occurred, that can be determined by the RAST. Very simple, friends. So always remember the significances only. RIST can be used to understand whether the IgE level is elevated than the normal or not. That's why we use the RIST. Simple point. Then the RAST technique will be used whether this IgE levels increased because of what allergen. So you have to remember, obviously the specific test is the RAST. Because if you only perform a RIST, you have to understand very correctly. If you only perform a RIST, what you understand only there is an increased level of IgE. You never know because of what allergen the IgE level is increased. So if you don't perform the RAST technique, RAST method, you can't treat that particular person, because without knowing we can't treat it. So that's why RAST is the most specific method when compared to the RIST. But it is very important, we have to perform the both tests in order to give the proper medication, in order to start the proper treatment. Look into the picture. I am talking about the RIST now. Okay, RIST, what we are reading? RIST, we are understanding only one thing. We are understanding whether the IgE level is increased or not. That's what I want to study. Now, friends, all the students follow me very correctly. Listen to me, friends. Here, this is, you can see, right? It's a paper disc or an agarose bead. Okay. So to the paper disc or agarose bead, what was attached? You can see here, there is a attachment of anti-IgE. Okay. You know what is this specialty of anti-IgE? It will go and attach, what function of anti-IgE is? It will always go and attaches to the patient IgE. Don't confuse, friends. Anti-IgE will come and attaches to the patient IgE. Okay. So first what we have done, we have taken the anti-IgE on the paper disc or an agarose bead. To that, from patient we have taken the blood sample. Now we know that that contains what? IgE level. Correct, ma? Now you only look into this. Now this anti-IgE will come and attaches to what, friends? Now that anti-IgE will come and attaches to the IgE. That is fine. Anti-IgE has come and attaches to the IgE. But what is our goal? Our goal is we have to study the level of the IgE. Suppose example, the IgE concentration is 0.2 grams per microliter. Example. 0.2 grams per microliter. Now, I want to know the level, whether the level of IgE is increased or not, or decreased. If it decreased, there is no allergic condition. If it is increased more than the normal, which indicates that there is a allergic. So I want to understand the level. So for that, what we will do, we will to this entire complex. What I am going to add? We are just adding the radio labeled anti-IgE. Now don't get confused. First, I have taken anti-IgE. To this, I have added patient IgE. Now the anti-IgE will come and attaches to the patient IgE. Now to these two, we are adding something. What we are adding, friends? We are adding radio labeled anti-IgE. So when we add the radio labeled anti-IgE, it will come and attaches to this complex. Now, you know, right, based on the color, here in the radio labeled techniques, based on the color, we can determine the level. Suppose example, I am saying, if the level of IgE is more than the normal, it will give one separate color. If the IgE level is less than the normal, it will give the different color. So by this, we can easily conclude whether the IgE level is enhanced in the body or not. So suppose example, if the IgE level is enhanced, so RIST is completed, which means we are clearly understood that there is a increased level of the IgE, we confirmed. But with only RIST we can't start the treatment. We have to use the method of RAST compulsory. Correct, no? Yes. So in order to use the RAST, very simple. Instead of the IgE, we will use the allergens. Very simple. Listen. We will use many allergens. Say example, I will use many allergens. See, I am using the grass. I am using some other dust. I am using certain animal dander. I will use different anti-allergens and I will treat with the IgE. Now, what I can observe one thing. I can observe because of what allergen this level is increasing. When suppose example grass allergen associated with the IgE, then its level is enhancing, which clearly indicating that the specific allergen is grass. Then we can start the treatment. So this can be proved by what, friends? RAST method. So if you want to do the treatment, it is compulsory. We have to use the RIST technique also, friends, as well as we have to use the RAST method also compulsory. Whatever we have studied in this 1 hour, that is only type 1 hypersensitivity. I don't know what you do. You have to remember the entire concept because everything is important. Now friends, if you look into the type 2 hypersensitivity, remember type 1 is very specific, caused by the elevated level of IgE. Concept is also very clear. When it comes to the type 2, what do you have to remember? Type 2 is exclusively caused by the antibody, and we did not mention, we did not specified it is caused by which specific antibody. That's why type 2 hypersensitivity we can call it generalized hypersensitivity. But the point is very clear, there is an involvement of the antibody. So whenever there is an involvement of antibody, it will do two functions, ADC, ADCC. In the morning class also I told you about ADCC. Okay, antibody dependent cell mediated cytotoxicity. That is called as, friends, ADCC. What is this ADCC? I have told to all of you, friends. If everyone listen. ADCC means what? Suppose example, cytotoxic T cell wants to work. Natural killer cell wants to work. It will be requiring the help of antibody. That's what we can call the antibody dependent cytotoxicity. Don't get confused with that particular point. And what's the point you have to remember here? Look into the diagram, everyone. Please don't get confused. In the type 2 hypersensitivity, this is the antibody, and everyone pay attention. This is the surface antigen. Now example, there is a complement molecule. Now the ultimate goal of the complement molecule is to destroy this. That is the ultimate goal. Now you know what is happening. Now this particular antibody first attaches to this, not the complement molecule. Complement molecule cannot attach to the antigen here directly. First antibody attaches to the antigen, then it stimulates the complement molecule, then the complement molecules, once it get activated, then it will destroy that antigen. So which means complement molecule, if it want to show any action, require the help of antibody. So that's why antibody dependent cell mediated cytotoxicity. Very simple it is. Okay, I hope it's clear until here. Okay, so that's all for today. Once again, I'll take the 2 classes in the tomorrow class also. Okay, so tomorrow is Monday. I will do like this only: Tuesday, Wednesday, Thursday, Friday. Okay, anyway, I don't teach anything on the Saturday. See, Monday, Tuesday, see, I will teach here unit 5, unit 6, unit 7, unit 8, unit 10. We will do this. I will teach minimum 3 topics for very sure in one single day, by giving the gaps. You do your generalized preparation without stopping it and attend this 1.5 hour class also. That's all enough. You don't need to take too much of stress at all. Okay, and prepare. And my last and final time I am saying to you, Monday to Friday. Please listen, keep it in your mind. At least, I'm not joking friends, at least read 10 to 13 hours minimum. If you can read more also, that's good, because the last 10 days whatever you study, brain will remember for a lot of time. And I never encourage any one of you to study the new topics. When you study the new topics, your confidence will be killed and you can't remember anything. Only read the topics which you have already studied, which you have already revised. Don't study any new topics at all. Okay friends, I'll see you